Welcome to the deep dive. Today, we're getting into something really fundamental. I mean, where did life even come from? It's a huge question. And, well, it turns out the story starts with chemistry.
That's absolutely right. This deep dive is all about chemical evolution. So how the actual elements, the basic stuff of the universe, had to come together and organize before biology could even get started. It's a real journey from stardust to cells.
And we're diving into the source material you've provided us. It's a great collection of excerpts walking us through this whole scientific story. So our mission today is to go through it really point by point, unpack the key ideas, and see how chemistry really set the stage for life, all the way up to searching for it elsewhere.
Yeah, it's a fascinating field because it connects, these massive cosmic events from billions of years ago right down to the chemistry inside a single cell. The material really shows how interconnected it all is. Our job is to help you navigate that, make those connections, maybe point out some of the surprises along the way.
Okay, let's do it. Let's unpack this story. And, well, where else can we start but right at the beginning? With the elements themselves.
Exactly. The first thing the material makes really clear is that the elements life needs, carbon, nitrogen, oxygen, phosphorus, sulfur, calcium, hydrogen. The big ones, they weren't just readily available right after the Big Bang.
Right, because the Big Bang mostly gave us hydrogen and helium and just a tiny bit of lithium that was, 13.8 billion years ago?
Precisely. And the sources really emphasize this. All the heavier elements, they had to be forged later, much later, inside stars, and then scattered across space when those stars died, especially in supernovae.
So element factories in space.
Basically, yes. And this element synthesis, it took billions of years. You absolutely needed those heavier elements for things like, water, you need oxygen for that, and solid planets, silicon, iron, magnesium, and eventually the building blocks of living matter. All that had to exist long before life could even, potentially form.
So the universe needed a really long cooking time just to get the basic ingredients together. Before scientists figured out this cosmic angle, what did people think about life's origins?
Well, the material touches on some of the older ideas. A really big one was spontaneous generation. Just the idea that life could sort of pop into existence from non-living stuff under everyday conditions.
Like maggots from meat, that kind of thing?
Exactly that kind of thing. And experiments by people like Redi, Spallanzani, and then Pasteur, famously, they disproved that idea. They showed that, generally speaking, life comes from pre-existing life.
And there was also vitalism, right? This idea that organic stuff, the molecules of life, could only be made by living things because of some special life force.
Right. That was another major hurdle. But that idea took a big hit back in the 1820s. Wöhler managed to synthesize urea, which is definitely an organic compound found in living things from purely inorganic chemicals in his lab.
So no vital force needed.
Exactly. It was a huge demonstration. It showed that organic chemistry follows the same rules as inorganic chemistry. It doesn't require a living organism to happen.
Which really paves the way for the idea of chemical evolution.
It does. Once you move past spontaneous generation and strict vitalism, the concept of chemical evolution starts to make sense. It's the idea of the natural, non-biological formation of the organic building blocks needed for life. The material points to some early experiments hinting at this, like Loeb's work around 1913 or Bodich's. They were synthesizing things like sugars from simple formaldehyde.
Wow, even back then.
And even earlier, Strecker, back in the mid-1800s, he showed you could make potential precursors to amino acids from really simple stuff like hydrogen cyanide, HEN.
Okay, here's something that really jumped out at me from the material is these organic building blocks. They aren't just an earth thing.
And this is, I think, one of the really mind-blowing points. Organic molecules, things like that hydrogen cyanide, HCN, but also methanol, formamide, quite a few others, they've actually been detected way out in interstellar gas clouds and meteorites that fall to Earth.
So they were out there before Earth even formed.
Exactly. That's what it implies. These crucial chemical precursors are older than our planet. They weren't necessarily cooked up on the early Earth. Some were likely delivered here. It suggests the basic ingredients for life might be, pretty common throughout the universe.
That definitely changes the picture. Earth wasn't necessarily special, maybe just lucky, right place, right time, right conditions.
That's one way to look at it.
Yeah.
So we have elements from stars, organic molecules from space. Where and when does the chemistry start getting complex enough to actually head towards something like life?
Where does the action happen?
The material suggests the best settings were probably within newly forming solar systems. Maybe during the process where planets or large moons were clumping together, but you needed specific environmental conditions.
Okay, what are those key conditions?
The sources consistently point to three main things interacting. First, an atmosphere providing the precursor gases, things like methane, ammonia, hydrogen sulfide, maybe carbon dioxide. Second, a solid surface. This is really important because reactions tend to happen much better when molecules can gather and concentrate on mineral surfaces, for example.
Instead of just floating around.
Right. And third, a liquid environment. Something like oceans or lakes acting as a solvent, letting things move around, meet, and react.
And I guess energy is crucial too, like sunlight, photochemistry.
Absolutely. Light, especially high-energy UV light from a young star, is a major energy source. The material explains how this photochemistry could really drive reactions. It could change the atmosphere's composition, maybe turning methane into CO or CO2. It affects liquids too. Light can break down water or ammonia or methanol if those were the solvents. Or, like on Saturn's moon Titan, it can cause hydrocarbons in lakes to link up, forming thick, oily sludges.
So light can build things up, but maybe break them down too.
It's definitely a double-edged sword. It provides energy for synthesis, but it can also destroy the complex molecules you just made if they're too exposed.
You mentioned solid surfaces being important for concentrating molecules. Is there any evidence for that idea from, say, Mars?
Yes, actually. The material brings up the findings from the Curiosity rover. Curiosity drilled into Martian rocks and found that the key non-metal elements, carbon, hydrogen, nitrogen, oxygen, phosphorus, sulfur, the CH and OPS elements, they're present in minerals like clays and gypsum. And these minerals clearly formed in water that was likely neutral, not too acidic or alkaline.
So early Mars had the right kind of watery, rocky environments.
It strongly suggests that yes, environments where these building blocks could potentially form and accumulate on surfaces.
And what about the idea that life might have started or survived hidden away below the surface?
That's another important point the material raises. Early planetary surfaces were probably pretty harsh places. Intense UV radiation, cosmic rays, maybe reactive chemicals called oxidants. Subsurface environments, pockets of water within the rock perhaps could have offered protection, more stable conditions, maybe a better place for complex chemistry or even early life to get a foothold and persist.
Which leads us nicely to the actual search for life on Mars. Specifically, those Viking experiments back in the 70s. That was the first time we really tried to look for active life metabolism on another planet, wasn't it?
It was incredibly ambitious for its time. Groundbreaking, really. The Viking landers had several experiments, including one called the Pyrolytic Release Experiment, or PRX. The basic idea was clever. Take a sample of Martian soil, give it some gas containing radioactive carbon-14, maybe as CO or CO2, and see if anything in the soil incorporates that labeled carbon into organic molecules. That would suggest metabolism.
Right, and the results, I know they've been debated ever since. What did the sources say?
Well, the material reflects that ongoing debate. It notes the PRX results were complex. Intriguing, but complex. And scientists had trouble fully reproducing them in labs on Earth using simulated Mars conditions. While some initially saw potential signs of microbial activity, the consensus reflected in the material leans more towards complex, non-biological chemical reactions in the Martian soil. Or at least the results remain ambiguous. It wasn't a clear yes.
It really highlights how hard it is to be sure you're seeing biology and not just some unusual chemistry, especially remotely.
It's a major challenge.
And this brings up a really fundamental distinction the material emphasizes.
Right.
Chemical evolution versus biogenesis. They're not the same thing.
No, definitely not. It's a critical point. Chemical evolution is the process we've been talking about. Simple inorganic stuff becoming more complex organic building blocks. Amino acids, nucleotides, sugars, lipids, that sort of thing, naturally. Biogenesis is the next, much bigger step. It's the transition from that collection of complex molecules to the very first systems that could actually replicate themselves, mutate, and undergo Darwinian selection.
So chemical evolution makes the parts list. Biogenesis builds the first machine.
That's a pretty good analogy, yes. Chemical evolution provides the necessary ingredients. Biogenesis is the spark, the origin of life itself from those ingredients.
And when we talk about making the ingredients, the most famous experiment has got to be milliurate.
Oh, absolutely seminal. The material rightly highlights the 1953 milliurate experiment. They tried to simulate conditions thought to exist on the early Earth. A mix of gases like methane, ammonia, hydrogen, water vapor, and zapped it with electrical sparks like lightning.
And
they successfully synthesized a variety of organic compounds, including several types of amino acids, which, as you know, are the building blocks of proteins. Now, the exact composition of Earth's early atmosphere is still debated. Maybe it wasn't exactly like their mixture. But the experiment was a massive proof of concept. It showed you don't need life to make the building blocks of life. They can arise naturally from simple chemistry.
Okay, so we've journeyed from stars creating elements to organic molecules forming in space, conditions needed on a planet, and this crucial difference between chemical evolution and actual biogenesis. Let's shift focus a bit. How do scientists try to understand which elements biology actually ended up using and why? The material introduces something called the biological system of the elements.
Right, this offers a different kind of perspective. It's called the BSE, the Biological System of Elements, developed by Markert starting in the 1990s. And its goal is different from the traditional periodic table that we all learned in school. The BSE tries to understand why certain elements are used by life, how organisms manage to selectively pull specific elements out of their environment, which is called fractionation, and then where those elements end up inside the organism. The material particularly focuses on how this works in plants.
Okay, so how is it different from the regular periodic table, the PSE?
Well, the periodic system, the PSE, organizes elements based on their atomic structure, protons, electron shells. And it's brilliant for predicting basic chemical behavior. But the material mentions some critique, citing folks like Allen, who argue the PSE has irregularities and maybe lacks a core organizing principle that's directly relevant for biology or geology. The BSE, on the other hand, tries to organize elements specifically based on how they behave within living systems.
Interesting. So what does the BSE look like? How is it structured?
It's usually shown as a kind of triangular diagram. The way elements are positioned relates to things like how their abundance correlates with other elements inside organisms, their tendency to form complex structures or polymers, and their overall role in biological functions. There's this really neat idea presented that different parts of a plant, leaves, roots, stems, can act almost like chemical ligands.
Ligands, like molecules that grab onto metals.
Exactly, as if the plant parts themselves are selectively binding and sorting elements they absorb from the soil and water, actively fractionating them.
That's a cool way to think about a plant, like it's doing chemistry to filter what it needs. What does this biological system tell us about what life is made of compared to, say, rocks?
A key point the material emphasizes is how different life's composition is. The bulk of living matter is overwhelmingly made of just a few non-metals: carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur, plus some key metals like potassium, calcium, magnesium. These are the macroelements needed in larger amounts. Then you have the microelements or trace elements, needed in much smaller quantities, but still absolutely vital. This elemental recipe for life is radically different from the Earth's crust, which is mostly oxygen, silicon, aluminum, and iron.
Life definitely cherry-picks its elements.
Very much so. It concentrates what it needs and excludes a lot of what's abundant geologically.
I bet studying those elements needed in really tiny trace amounts is super difficult experimentally.
Oh, incredibly difficult. The material discusses this challenge. Proving that an element is truly essential when it's only needed in, say, parts per billion amounts is tough. Why? Because it's almost impossible to eliminate that tiny amount from the environment in an experiment. Contamination from lab equipment, dust in the air, even impurities in the pure water or nutrients you're using can accidentally provide enough of the element for the organism.
So you might think it's not essential, but it was there all along.
Exactly. It makes definitive proof for some ultratrace elements very tricky.
So how do researchers use this BSE approach to compare different types of plants?
The material talks about the reference plant concept, which Markert also developed. It's basically an averaged elemental composition, a standard baseline. Scientists can then analyze a specific plant species and compare its chemical fingerprint to this reference. It helps standardize comparisons. And what they find, the material notes, is that plants that are genetically related often have quite similar patterns of element distribution, which suggests strong biological control over element uptake and allocation, not just passive absorption.
Does this analysis reveal any interesting connections or correlations between different elements in plants?
Yes, the material gives a few examples. Potassium, for instance, tends to correlate well with most other macro elements and also the halogens, which fits its role as a key ion for maintaining charge balance and osmotic pressure. Aluminum, iron, and scandium often track together with the rare earth elements, the lanthanides, probably because their ions have similar sizes and charges. And they mention high correlations between boron, phosphorus, and nitrogen. This provides circumstantial evidence supporting boron's essentiality in plants, even though pinning down its exact biochemical job has been surprisingly difficult.
So the BSE provides this biological viewpoint. The material also briefly mentions a third system, the geochemical system.
Right, just briefly, the geochemical system of the elements, or GSE, attributed to Railsback. This system focuses on how elements behave in geological settings, how they partition between different minerals, rocks, water, over geological timescales. The material includes a useful table comparing the three systems, PSE, GSE, and BSE.
How do they compare?
Well, the PSE is ancient, deals with the whole universe, and focuses on atoms. The GSE is geological timescale, focuses on Earth's layers, and deals with ions and minerals. The BSE is much more recent, focuses on biomass, and deals with elements within organisms. Three different ways of looking at the same elements, depending on the context.
It's fascinating how the same basic ingredients can be organized and behave so differently in a star, a rock, or a cell.
It really is. And this ties back nicely to the idea of element evolution, or, as Williams and Frosta da Silva termed it, the natural selection of chemical elements. It's not biological selection in the Darwinian sense, but the idea that the specific set of elements life uses today is a result of a long history. It reflects what was available in the environment when life arose and evolved, and the functions that needed to be performed. Big evolutionary steps, like the origin of eukaryotes through endosymbiosis, actually increased the number and types of elements that became essential.
Organisms basically adapted their internal chemistry based on what was outside and what jobs needed doing. Okay, let's dive deeper now into those specific jobs, analyzing the biological roles of chemical species.
Right, and this chapter starts with a really fundamental principle, echoing Paracelsus from centuries ago. An element's effect depends on more than just if it's present. It depends on its chemical form, its speciation, and critically, the amount, the dose. Too little can be bad, but too much can also be bad. The dose makes the poison, as they say.
And the material highlights some roles for elements that are, frankly, pretty surprising, stuff you wouldn't expect to be essential.
It really does. Take cadmium, for example. We usually think of cadmium as a toxic heavy metal, but the material points to a specific marine diatom, a type of phytoplankton called Thalassiocera weissflogii. For this organism, cadmium is actually required under certain conditions. It can substitute for zinc in an important enzyme called carbonic anhydrase.
Cadmium. Essential. Wow, okay.
Then there's barium. The material mentions certain types of algae, desmids, that live in very nutrient-poor acidic bogs. They seem to require barium for growth. But remarkably, the sources state its actual biochemical function. What it does is still unknown.
So it's needed, but we don't know why.
Pretty much. And then there's tungsten, element 74.
Tungsten, like in old filament light bulbs. Essential.
Believe it or not, yes. The material highlights it as the heaviest element known to be essential for any form of life. It's used in certain enzymes, specifically types of oxidoreductases found in some very ancient microorganisms, archaea, and bacteria like Clostridia. Often these organisms live in extreme environments, like volcanic vents, very high temperatures, or environments with very little oxygen, very low redox potential. Under those conditions, tungsten works in enzymes where a similar element like molybdenum, which is more commonly used, apparently wouldn't function properly.
That's amazing. What about lithium? We use lithium salts in medicine for treating bipolar disorder. Is it essential for life?
Lithium. According to the material, lithium is a real puzzle. Yes, it's used medically, but the sources are quite explicit, citing specific researchers that, as of their writing, there is no definitively known required physiological function for lithium in any living organism. None is proven essential.
Right. Despite the medical use.
Right. They mention hypotheses for how it might work therapeutically, like the inositol depletion hypothesis, but that doesn't mean it's required for normal, healthy life. And of course, lithium has toxicity issues at higher concentrations.
So even for an element we use as a drug, its fundamental biological role is a mystery. That's quite something.
It is, and the material acknowledges this broader point. For many elements, even ones we know are required for growth or reproduction, the precise molecular job they do isn't fully understood. It also brings in the idea of autocatalysis again, those self-accelerating reactions, suggesting that the principles of autocatalytic network theory might help explain why life settled on a relatively constrained set of elements for its core catalytic machinery. It didn't just grab everything available.
Beyond individual elements, how can looking at the overall pattern of elements in an organism tell us something about Earth's deep past, or evolution?
This is where it gets really cool, linking present-day biology to ancient environments. The material uses the example of amphibian metamorphosis, how tadpoles turn into frogs. This process absolutely requires the hormone thyroxine, which contains iodine.
Right.
But if tadpoles live in an environment that's very low in iodine, some species have evolved the ability to stay as tadpoles their whole lives, even reproducing in that larval state. It's called neoteny. The material speculates that this biological phenomenon might be a kind of chemical memory. Maybe it reflects conditions deep in evolutionary history, perhaps when early tetrapods were evolving, where iodine, or maybe even copper, which is also involved in hormone processing, was scarce in certain environments.
So the biology we see today could hold clues about ancient geochemistry.
Exactly. The idea is that by studying the details, maybe the specific metal-using proteins, metalloproteins, in modern tadpoles that retain this ability, we could potentially get hints about those ancient environmental conditions.
That's fascinating detective work. Are there other extreme examples in the material?
Yes, there's a really striking one, the loriciferans. These are tiny marine animals. The material describes some found living in incredibly harsh conditions, deep Mediterranean brine pools that are completely anoxic, no oxygen, and hypersaline, super salty. Compared to their relatives living in normal oxygenated seawater, these anaerobic loriciferans have a dramatically different elemental makeup.
How different?
They seem to completely lack magnesium and iron, which are essential for most life. Instead, they have much higher levels of copper and also an enigmatic need for bromine. This isn't just a small adjustment. It implies a radical overhaul of their entire metabolism to survive without oxygen and with this weird chemistry. For example, iron is often involved in anaerobic respiration pathways like sulfate reduction, so lacking iron means they must do things very differently. Maybe lower phosphorus levels indicate a slower overall metabolism too. Their elemental fingerprint is a direct result of adapting to that extreme niche.
Wow. It really shows how flexible life can be in its chemical requirements when pushed to the limit. And you mentioned changing environments earlier. The material talks about big geological events that really changed which elements were available on Earth's surface, right?
Absolutely. Two huge ones are mentioned. First, the Great Oxygenation Event, roughly 2.5 to 3 billion years ago. When cyanobacteria started pumping out massive amounts of oxygen as a waste product of photosynthesis, it fundamentally changed Earth's surface chemistry.
How so?
Before widespread oxygen, many elements were dissolved in the oceans in a reduced state, iron, for example. But when oxygen levels rose, these elements oxidized and became much less soluble, precipitating out of the water, forming things like the banded iron formations. It also meant oxygen could attack sulfide minerals on land, releasing other elements. The material even includes a picture from Cyprus showing this kind of sulfide oxidation happening today. It basically rusted the planet.
And the other big factor was plate tectonics.
Yes. The start of plate tectonics, maybe as early as 3.7 to 3.9 billion years ago, according to the material, began recycling Earth's crust. This process drags surface rocks, including certain types of metal deposits, down into the mantle, changing the long-term availability of elements at the surface.
And mass extinctions, did they affect element availability?
Indirectly, yes. The material mentions the Ordovician and Permian mass extinctions, noting they might have been even more devastating than the KT event that killed the dinosaurs. While the extinction itself doesn't change elements, the causes often do. For example, the KT impact threw up dust rich in elements like iridium and osmium, which are rare in the crust but more common in asteroids. That's how we identify impact layers. Huge volcanic eruptions, another potential cause of mass extinctions, can release massive amounts of gases like sulfur dioxide, leading to acid rain, which then leaches metals out of rocks and soils, drastically changing water chemistry.
So the takeaway is that element essentiality isn't some fixed list. It has changed over evolutionary time and differs between groups of organisms.
Precisely. The material makes that point clearly. Think about the difference between the Precambrian era, dominated by microbes, and the Cambrian explosion, starting around 541 million years ago. That's when we see the rapid diversification of complex, multicellular animals. A key development then was biomineralization, the ability to make hard parts, shells, skeletons, teeth.
Right, like shells and bones.
Exactly. And interestingly, the material notes that some of the earliest hard parts weren't calcium carbonate like most shells today. Early groups like conodonts used calcium phosphate for their tooth-like structures. Vertebrates and echinoderms also used phosphate-based minerals like hydroxyapatite early on. Making these mineralized structures required organisms to evolve sophisticated biological machinery to actively control and process large amounts of metals, like calcium.
Can we trace this history using isotopes? You mentioned isotopic fractionation earlier.
Yes, that's another powerful technique the material mentions. Biological processes often slightly prefer one stable isotope of an element over another. For instance, photosynthesis prefers lighter carbon-12 over heavier carbon-13. By measuring the ratio of these stable isotopes in ancient rocks, organic matter, or fossils for elements like carbon, nitrogen, calcium, iron, zinc, even uranium, scientists can reconstruct past biological processes, figure out ancient diets, or track changes in environmental conditions. These isotopic signatures are like faint chemical echoes of past life.
It's incredible the stories you can read from these subtle chemical variations. Okay, one more point from this section. As animals got bigger during the Cambrian, they faced a new problem getting oxygen inside their bodies. How did element chemistry help solve that?
That's a great point. Once organisms got too large for simple diffusion of oxygen through their skin to supply all their tissues, especially if they developed gills or other barriers, they needed a way to transport oxygen internally. The material explains this evolutionary pressure led to the development of specialized oxygen-carrying molecules in the blood or equivalent fluids of early animals. And these molecules are based on metals, usually either copper, as in hemocyanin found in mollusks and arthropods, or iron, as in the hemoglobin we have.
So the need for efficient oxygen transport drove the biological recruitment of copper and iron for this critical job.
Exactly. It's another example of biology co-opting available elements for new, essential functions.
The material then draws an interesting comparison between catalysts, things that speed up reactions in different realms, prebiotic chemistry, modern biology, and industrial organic chemistry. What's the point there?
It's quite revealing. The material notes, using a table for comparison, that some elements that are actually pretty good catalysts in laboratory simulations of prebiotic chemistry elements like aluminum, titanium, zirconium, indium, maybe even lead, are conspicuously not used as major catalysts in modern biochemistry.
Why not?
The suggestion in the material is that it might be due to the different reaction conditions or the types of molecules involved. Prebiotic chemistry might have involved different solvents or occurred on mineral surfaces under conditions very different from the watery interior of a cell, often constrained within lipid membranes. Perhaps those elements are good at catalyzing reactions that aren't central to modern metabolism, or maybe they just weren't available in the right form, or perhaps they were excluded for other reasons, like toxicity.
Which brings us back to toxicity. How does that link to catalysis and binding?
The material reiterates a key principle. Toxicity is often related to binding strength, elements that form extremely stable bonds with essential biological molecules. The material gives examples like beryllium ion, B2+, palladium, PD2+, mercury, Hg2+, the uranyl ion, UO22+, plutonium, Q4+, tend to be very toxic. They essentially gum up the works by binding too tightly, often irreversibly, to proteins or DNA where they shouldn't, displacing essential metals or disrupting structure and function. There's that Goldilocks zone for essential elements. Binding needs to be strong enough to work, but not so strong that the element can't be released or managed by the cell.
That makes sense. You need control. Okay, that's a really thorough look at the roles of elements. Now, the material introduces a specific analytical tool. Stoichiometric network analysis, or SNA, what is that about?
Right, SNA. It was developed primarily by Bruce Clark starting in the 1970s. At its core, SNA is a mathematical framework specifically designed to analyze the dynamics of complex reaction networks, particularly those that exhibit autocatalysis.
Autocatalysis, that means.
It means a process where one of the products of a reaction acts as a catalyst for that same reaction. So the more product you make, the faster the reaction goes. It creates a positive feedback loop, causing the system to accelerate.
Okay, I can see how that might be relevant to life, which is all about growth and reproduction.
Exactly. The material explicitly connects SNA to understanding chemical evolution and also modern bio-inorganic chemistry. It's a tool that can help researchers analyze the complex web of reactions involving, say, metal-containing enzymes, metalloproteins. It helps understand how feedback loops within these biological networks influence whether an element is essential or potentially toxic.
Where else do we see autocatalysis in biology, apart from the big picture of reproduction?
The material gives a couple of examples. Think about the very beginning of yeast fermentation. The yeast cells produce enzymes that break down sugar, and making more enzymes allows them to break down sugar even faster, leading to that initial rapid burst of activity. Or imagine an organism encountering a brand new food source. The initial slow adaptation might lead to producing enzymes to exploit it, which allows faster growth, leading to more enzymes and so on. A rapid bloom.
A runaway reaction, in a sense.
In a sense, yes, although the material also points out that biological autocatalysis, like population growth, is always eventually limited by external factors, running out of food, energy, space, or being eaten by something else.
Does this kind of network analysis apply to whole ecosystems too?
The material touches on that briefly. You can think about trophic levels in a food web or the flow of biomass and energy through an ecosystem using similar network concepts. It mentions the mobile cave ecosystem in Romania as an interesting case study. It's a sealed cave system where the entire food web is based not on sunlight, but on chemotrophic bacteria microbes that get energy from chemical reactions, like oxidizing hydrogen sulfide. Understanding the cycling of essential elements within that unique chemical network is crucial.
There's a mention here of Vernadsky's idea again.
Yes, Vladimir Vernadsky, the great Russian geochemist, he had this grand vision that life, living matter, would progressively organize and activate essentially all the chemical elements available in the biosphere. The material offers a slightly sobering perspective on that, suggesting that, well, life hasn't actually gotten very far with that grand project just yet, at least not in terms of creating really strong, globally significant biological cycles for every element. The elements with the most dominant biological cycles, according to the material, are phosphorus and zinc, and maybe copper to a somewhat lesser extent. For many other elements, geological cycles are still much more dominant than biological ones on a planetary scale.
Okay. How might SNA help predict if a particular metal ion is going to be useful, essential, or harmful, toxic to an organism?
This is a potentially powerful application. The material discusses using SNA, combined with understanding the chemical properties of metal ions, to model their interactions within biological systems. The idea of the window of essentiality comes up again, visualized perhaps in a diagram like the one mentioned. Figure 4.5. A key concept here is binding affinity. Metals that form extremely stable complexes with biological ligands might actually be excluded from essential roles.
Because they bind too tightly.
Precisely. They might be difficult for the cell to manage, transport, or release, where needed for dynamic processes like catalysis. They just lock up and don't let go. So being too stable can be just as bad as being too weak or unstable. And researchers use various techniques like electrochemical methods and concepts like the electrochemical ligand parameter, ELL, to quantify these metal ligand binding strengths. Understanding how strongly a metal binds to different sites in proteins or other biomolecules is fundamental to predicting whether it's likely to integrate usefully or just cause disruption.
Does this analysis connect back to the very beginnings, to prebiotic chemistry?
It seems to, yes. One interesting point the material raises is that the set of chemical elements that experimentally turn out to be good catalysts in those simplified early-stage prebiotic reaction simulations we discussed earlier, well, that set looks remarkably similar to the core set of essential trace elements used in biological catalysis today. SNA provides a theoretical lens to explore why this continuity might exist. Perhaps certain metal ions just have inherent properties that make them particularly well suited for facilitating the kinds of reactions needed in autocatalytic networks, whether those networks are purely chemical or fully biological.
It's like the chemical logic that worked before life began was carried forward into life itself. A really powerful idea. Okay, let's pull back a bit and bring the environment back into the picture. Chapter 5 talks about the significance of water, soil, and atmosphere.
Yes, and the core message here is really about the interaction between these different environmental compartments. Chemical evolution, and certainly life as we know it, doesn't happen in isolation. It requires the interplay between a liquid phase for transport, dissolving things, allowing molecules to meet, a solid phase providing surfaces for concentration, catalysis, and chemical exchange, and an atmosphere acting as a reservoir for key volatile elements like carbon, nitrogen, sulfur, and also being shaped by energy inputs like light.
Water is obviously front and center as the liquid.
Absolutely. Water's unique properties as a solvent are crucial. But here's another one of those surprising nuances from the material. It suggests that maybe huge, almost unlimited bodies of water, like a global ocean, might not have been the ideal place for chemical evolution to get started.
Really? Not the primordial soup in the ocean.
The argument hinted at is that extreme dilution could be a problem. It might be hard to get molecules concentrated enough to react effectively. The material floats the idea that perhaps a little less water, maybe concentrated in smaller pools, ponds, or lagoons that could experience cycles of wetting and drying, might have been more conducive. Those cycles could naturally concentrate solutes as water evaporates, driving polymerization reactions or other complex chemistry that wouldn't happen in a dilute ocean.
That makes sense. Concentration is key. Evaporation could help.
It's one possibility. The material also briefly acknowledges that scientists consider other potential liquids as solvents for life elsewhere, things like liquid ammonia, methanol, formamide, or even the liquid hydrocarbons we see on Titan. Although the sources we're discussing focus overwhelmingly on water as the basis for Earth's life.
Okay, and the atmosphere, we touched on this, but its role.
The material reiterates its importance as the source of those key volatile precursors: methane, ammonia, water vapor, maybe CO2, hydrogen sulfide. And again, the role of photochemistry driven by sunlight, breaking down molecules and providing energy for new reactions. But it also emphasizes the need for geochemical constraints. We can't just assume any atmosphere. We look at ancient rocks, like the oxidation state of iron minerals, to deduce what the early atmosphere could have been like. For example, evidence from old rocks argues against a very methane-rich atmosphere extremely early in Earth's history.
And the solid part, the soil, the rocks.
Crucial for interaction. The contact between minerals and the atmosphere or water is where a lot of important chemistry happens. Exchange of elements, and critically, minerals can act as catalysts. The material revisits the Mars example here. The observation that organic compounds seem to rapidly oxidize on the Martian surface suggests the presence of strong oxidants, or catalytic minerals, in the soil and rock. Perhaps things like manganese oxides driving these reactions.
So it's the whole system, liquid, solid, gas, energy, all interacting.
Exactly. And the material wraps this up by reminding us that the fundamental physical conditions of a planet, its size, which determines gravity and its ability to hold an atmosphere, its distance from its star, which sets temperature and liquid water potential. These factors define the basic habitat limitations, the boundaries within which chemical evolution and life might be possible.
Okay, bringing all these threads together, the elements, the chemistry, the environments, how does this understanding guide our actual search for life beyond Earth? This leads us to the final chapter in the material: present and future projects on chemical evolution by means of space research.
Right. The core idea here is that space missions are absolutely vital. They let us test our Earth-based understanding in different settings and directly search for signs of chemical evolution or even life itself elsewhere.
Where are the main efforts focused right now?
The key targets highlighted in the material are, first, Mars. We've discussed the ongoing rover missions like Curiosity, which are meticulously examining Martian geology and chemistry for signs of past habitability. Second, the icy moons of the outer planets are incredibly exciting, particularly Saturn's moons Titan and Enceladus and Jupiter's moon Europa.
Why those moons?
Well, Titan has that thick atmosphere and incredibly complex organic chemistry happening on its surface, albeit at frigid temperatures. Enceladus and Europa are prime candidates because they almost certainly have vast liquid water oceans hidden beneath their icy shells. Exoplanets orbiting other stars are mentioned too, but they're just so far away that detailed chemical analysis is extremely challenging for now, though atmospheric spectroscopy is making amazing progress.
Can you give some specific examples of what we've learned from missions?
Sure. On Mars, as we said, Curiosity's drilling and onboard lab have confirmed the presence of ancient lakebed environments with clays, sulfates, and all the key nonmetal elements needed for life as we know it. It paints a picture of a potentially habitable early Mars.
And Titan.
The Cassini mission, with the Huygens probe that landed on Titan, gave us incredible insights. It analyzed the composition of the atmospheric haze and even droplets during descent, finding complex organic molecules like hydrogen cyanide, HCN, cyanoacetylene, HC3N, and many others. It's a natural laboratory for complex organic synthesis in the absence of liquid water at cryogenic temperatures.
What about the ocean moons, Enceladus and Europa? The potential for life there seems huge.
It's incredibly tantalizing. The material highlights the Cassini mission's discovery at Enceladus geysers, plumes of water, ice, and vapor erupting from cracks in the South Pole, spewing material from the subsurface ocean directly into space.
Wow, so we can sample the ocean without drilling.
Exactly. Cassini flew through those plumes and detected water, salts, silica nanoparticles, suggesting hot water interacting with rock at the ocean floor, and even complex organic molecules. It's a direct window into a potentially habitable environment. Europa also has strong evidence for plumes, though they seem less frequent or smaller, making sampling harder. And drilling through Europa's potentially much thicker ice shell is a massive technological challenge for the future.
Did the material mention Triton, Neptune's moon?
Yes, briefly. Voyager 2's flyby revealed Triton's another fascinating cryogenic world with nitrogen ice, a thin nitrogen and carbon monoxide atmosphere, and signs of active geology and potentially organic chemistry, too.
Have we brought any samples back from these distant places besides meteorites?
Yes, the Stardust mission is mentioned specifically. It flew through the coma of Comet Wild 2, collected dust particles on a special gel, and returned them to Earth. The material emphasizes how valuable these samples are because they represent incredibly old, primitive material, potentially dating from before the solar system formed, giving us a direct look at the starting materials for chemical evolution.
Looking ahead, what are the prospects? Are missions like drilling on Europa feasible?
The material acknowledges the immense challenges cost, complexity, and especially avoiding contaminating these pristine environments with Earth microbes. Drilling on Europa is seen as a long-term goal, very difficult, but potentially revolutionary. In the nearer term, missions will continue exploring Mars, potentially returning samples, and planning missions like Europa Clipper to do detailed reconnaissance of Europa's ocean and plumes. The New Horizons mission's flyby of Pluto and Charon is also noted as expanding our knowledge of these distant icy bodies in the Kuiper Belt.
So, based on everything we've discussed, the ongoing work, the future plans, do the sources offer a prediction? When might we actually find life elsewhere if it's out there in our solar system?
The material concludes with a sense of, I'd say, cautious optimism. It suggests that given the pace of discovery, the technological advancements, and the missions currently planned or envisioned, the detection of extraterrestrial life forms within our solar system, if they exist, seems potentially achievable within the next few decades.
Wow, okay. That was quite the journey, a real deep dive. We went from the Big Bang and element creation in stars through the first chemical steps on early planets, looked at how life intricately manages elements using systems like the BSE, explored tools like SNA to understand these networks, considered the vital roles of water, rock, and air, and landed on the ongoing search for these very processes on worlds like Mars, Titan, and Europa.
It really is a story that unfolds layer by layer, from simple chemistry to complex biology, all shaped by the elements available and the conditions provided by the environment. And as the material showed, studying how different organisms here on Earth have adapted their use of elements, sometimes in really surprising ways, like that cadmium using diatom or the luciferins, it really makes you wonder about the sheer range of chemical possibilities life might employ elsewhere.
Absolutely. And remember, this deep dive was put together specifically for you, using the detailed source material you provided to give you this focused exploration of chemical evolution and the origins of life.
So here's something to maybe leave you thinking about.
Given how life on Earth demonstrates this chemical flexibility, adapting its elemental needs sometimes radically to fit different niches, what kind of truly exotic chemical requirements might life evolve on a world like Titan, with its methane lakes, or Europa with its potentially salty dark ocean? And if we could figure that out, what would it tell us about the truly fundamental, maybe universal, requirements for what it takes to be alive anywhere in the cosmos?
That's the big question. Thank you for giving us such fascinating material to explore together today.